are we looking for? Mycobacterium, tuberculosis, or leprae. Or nocardia would be an option. Okie dokie. Okay. When we did our acid-fast stain, if the cells came out green, what does that mean? It's acid-fast negative, or I call it non-acid-fast. Well, what does that mean?
That they are not acid fast.
Right, they're not acid-fast. They do not have the wax in their walls. What's the name of the wax? Mycolic acid. So if it's non-acid-fast or acid-fast negative, it does not have the waxes in the wall. So, what was the example of our non-acid-fast bacteria on our AFP slide? E. coli. E. coli, good. What color is it going to come out if it does have the waxes?
Red.
Reddish, it's a pink. It's technically pink. On your test, I'll use the word pink. But some books will say red. Okie dokie. When you do a Gram stain, we just did this in lecture today. What if the bacteria come out pale red?
Then it's gram positive.
No?
Gram negative.
Yes. And what does that mean if it's Gram negative?
That it has
Endotoxins?
yes.
And a thin wall. Now, Gram-positive bacteria can actually have exotoxins and endotoxins. Gram-negative bacteria only have endotoxins. I know it's a little weird and confusing, but just keep in mind, Gram-negative, that's endotoxins. Gram-positive, they have exotoxins, but they can also have endotoxins. Okay, let's see. Okay. So, what if in a Gram stain it comes out purple?
Gram positive.
Gram positive. Good. Thick wall. Okay. What do you know about E. coli?
Green.
It's division 1, so it has a thin wall, right? It's enterobacteria, comes from your digestive tract. That's where it gets the name coli. It's also non-acid fast, so it's going to come out green if you do an acid fast stain. And what color is it going to come out if you do a Gram stain? Red. And what's the shape of the cells? Broad shape, tiny bacilli, yep. And if you see it on a Petri plate, what color is it going to be?
Like
Whitish. It's going to be white on the Petri plate. Not white, opaque, bright white. It's going to be like a creamy, translucent white. Okay. What if I asked you, if I did a Gram stain of Staphylococcus or Streptococcus or Micrococcus or Enterococcus, what color is it going to come out?
Red. No, purple.
Purple. Why?
Gram
They're
positive.
Gram positive, right? Gram positive cocci. Cocci or Gram positive, they're going to come out purple. Good. How about our 2 endospore forming bacteria, Bacillus and Clostridium? And Lactobacillus. They're thick-walled, larger bacilli. They're also Gram positive. How about your little rods like Escherichia coli and Enterobacter and Shigella and Salmonella and bacteria in the family Enterobacteriaceae? They're division 1 Gram negative thin wall. They're tiny bacilli. I'll get you some props in a minute. Okay? Don't forget your shapes and arrangements of bacteria. Streptococcus, Staphylococcus, Diplococcus, Diplobacillus, Streptobacillus. There's single bacilli, there's cocci, there's spirillums and spirochetes, right? Okay? Okay. Don't forget the cellular morphology and the colonial morphology. Right? I told you to look them up. Need to know the cellular morphology. That's the shape and arrangements of the cells for Staphylococcus epidermidis, Micrococcus luteus, Bacillus subtilis, and Escherichia coli, and Serratia marcescens, because those are the 5 we've looked at in lab so far. A lot of times you'll get a question about what's the cellular morphology, what's the colonial morphology. The answer is usually easier than you think. You're thinking, oh crap, I didn't memorize the cellular and colonial morphology of all 5 of those bacteria. I'm never going to remember. The answer is in the name. If it's coccus, it's round-shaped. If it's Staphylococcus, they're round-shaped in clusters, right? If it's luteus, it's yellow. Remember, Serratia was supposed to be red pigmented? So on the Petri plate, it would have red colonies. Yours just started to produce red. It wasn't completely red. So a lot of times, the answer's already there. It's not too hard to figure out. Remember, Serratia are the red bacteria. Micrococcus luteus are yellow. Staphylococcus aureus, aureus means gold. And you saw some of them on your Petri plate. Okay, did that one. Well, I think I have the same question in there twice. With different options for the answer, huh? Okay. Remember, we talked about fermentation. And when Dr. Pasteur was looking at wine, if the grape juice turned to wine, that was alcohol fermentation. If the wine turned to vinegar, that was acetic acid fermentation. In gangrene, the gas gangrene gives off butyric acid. That's butyric acid fermentation. When we looked at yogurt, the milk, lactose, turns to yogurt, which is tangy. That's lactic acid fermentation. You also have lactic acid fermentation in your sore muscles, right? And, okay, remember which stains are basic and which are acidic? Purple, blue, red, and green. Crystal violet, methylene blue, safranin is red, brilliant green. Those are basic dyes. Negative stains, nigrosin, which is India ink, black, Congo red, and eosin. Those are your negative stains with the acidic charge. Okay, you got to read your documents that are in Canvas, because sometimes there's questions on them that I'm going to ask on the quiz. Do you know what prebiotics are? Do you know what probiotics are?
Yeah, I'm not gonna use the vacuum.
Probiotic is healthy bacteria. You want healthy bacteria on your body, in your body, in particular in digestive tract. So foods like yogurt that are naturally made by fermentation or even manufactured, they have healthy probiotic bacteria in them that contribute to a good healthy flora in your body. So they actually, to some foods, add extra probiotic bacteria. But these probiotic bacteria, they need to hang out. They need fibers to hang out on. Remember the beer chips I showed you? They throw those strips of beech wood in the beer containers. Why? So the probiotic yeast, the yeast that ferments the beer, has something to hang out on, something to cling to. Same thing with prebiotics. They're fibers. You need to have fibers in your digestive tract for your probiotics to hang out on. So some foods add prebiotics also. You especially see it in dog food for some reason. They add probiotics and prebiotics to dog food. They're giving the dogs fiber and healthy bacteria, so the healthy bacteria have something to live on in the digestive tract. That's the prebiotic, is the fiber. Okay, you know what commensals are? That's your good guys. You live together peacefully, mutually beneficially, commensal. As opposed to your pathogenic bacteria, which are the ones that might have an opportunistic infection, gets out of control. Your commensal turns into a pathogen, so to speak, or pathogens that come from your environment that make you sick. Okay, why do we even stain our bacteria? Make it easier to see them, right? We can see the shape and arrangement of the cells because we need contrast, so we stain to see contrast. Makes it easier to see the cells in our microscope if they have better contrast. That's one of the things that improves resolution. If we can see the shape and arrangement of the cells in our microscope, it helps us identify the bacteria, right? Helps us see the cellular morphology better. Okay. Oh, yeah. Oh, you know what? I might not have mentioned this yet. Let's do this. Penicillin targets what?
Cell wall.
The cell wall. What specific component of the cell wall? The peptidoglycan. So if I'm gram-positive cell and I have lots of peptidoglycan, or I'm a gram-negative cell and I only have little peptidoglycan, plus it's covered by that outer membrane, who do you think is going to be harmed more? What?
The positive.
The gram-positive bacteria. Penicillin harms gram-positive bacteria more than gram-negative bacteria. In fact, most gram-negative bacteria are resistant to penicillin because the little bit of peptidoglycan they have is covered by an outer membrane. But if you're a gram-positive cell and all you got is peptidoglycan and a lot of it, and it can damage a lot of your peptidoglycan, you're really vulnerable to penicillin. So penicillin gets gram-positive cells much, much better. It's meant for gram-positive bacteria, not gram-negative. And remember who's gram-positive. All your cocci and both of your endospore formers. Bacillus and Clostridium are your endospore formers, they're gram-positive. And all cocci are also gram-positive, so they're going to be susceptible to penicillin because of that thick wall of peptidoglycan. Oh boy. Okay, got to know your shapes and arrangements of the species and a little bit about cellular morphology. You need to know the two genera that are endospore formers. Where do endospore forming bacteria come from? The dirt, right, they're in the soil. Can you name a spirochete? What? Syphilis, good. What's the scientific name for syphilis? Remember that one. Treponema pallidum. There's another spirochete causes Lyme disease. What's the name of that?
Borrelia.
Borrelia burgdorferi. Good. Okay. Sometimes you don't know the answer to the question, but you do know what it's not. So read the question. Look at A. Could it possibly be A, or there's no way it could be A? Cross it out. Look at B. Does B answer the question, or is there no way? No way. Cross it out. So sometimes you don't know the answer, but you know which ones are not the answer. So figure it out by process of elimination. Let's say you eliminate 3 of the choices and you're down to 2. Well, that's better odds than you had before. Guess one of the 2. You see what I mean? Okay. Do you want me to write the species of the bacteria that you need to know on the board?
Yes.
I'll put them here.